So hello again. Now I'm going to walk you through two important documents. First of all, the ILO document revisited. Here, as you can see, I have added two columns. One column showing the ILOs that are expected to appear in your end on your end of module exam, and the second column showing the ILOs that are expected to appear on your end of year exam. I have additionally highlighted some ILOs because I thought, well, I want you to study very well and I want you to get the highest grades possible. So um, these ones highlighted in yellow, I think that the questions that might target these ILOs will be a bit complex, a bit deep. So I want you to study these really well and to look into um, the depth of the concept and to understand them really from the basics. You will see that some ILOs are marked for both exams, which means they might be covered in any of these exams, and some ILOs are not even marked at all. And this is just a gift. Okay, that this means that these ILOs that are not marked will not appear on your exam this year. This is not a strike through. This is a problem with the document that I cannot get rid of. So please, this is not canceled. It's simply a mark. As you can see here, certain ILOs are marked in yellow. You need to take really good care of these. And again, regarding the drugs used in osteoporosis, I have outlined before and mentioned that osteoporosis is a very bad disease because it does not affect just one individual. It affects the whole family and the community because it turns someone from a productive person, if that person has a fracture or develops a fracture, or that person will become let's say bedridden, or will become dependent on others. That's why these drugs are very important. Please take care of two things, actually of three things. Any cautions associated with these drugs and their uses, mechanism of action in details, and the selection of each drug type in each particular situation. So this is going to be really important, these osteoporosis medications. Again, I know this part you don't like, which is the part with so many drug names, the noradrenaline, serotonin reuptake inhibitors, tetracyclic and unicyclic antidepressants, serotonin receptor modulators. So many names here. This is going to appear on your end of year exam. Regarding the drug names that you need to know, I'm going to refer to the drug list now, and hopefully it's going to be clear enough. Common hallucinogens, it's not about the names of the actual drugs. It's about the mechanism of action at the cellular level and how this relates to the effects they have on a particular individual. But again, this is an important topic. Important side effects of antipsychotics, you need to know the actual mechanism of each side effect. Now, classifying narcotic analgesics, this is a bit tricky because here, and only here, I need you to know the chemistry. I need you to know which compounds are natural, which are semi-synthetic, which are synthetic, which compounds are pure agonists, which are pure antagonists, which are agonists, antagonists, which opium alkaloids, okay, like phenanthrene and non-phenanthrene alkaloids. Which ones are narcotic? Which ones don't even have narcotic effects? This part I need you to know very well. The therapeutic uses of opioid antagonists, you can just take my word, and this page or half a page, just know it by heart, please. Again, the individual non-steroidal anti-inflammatory drugs in the last orientation or revision, I told you that this is very important because non-steroidals are there everywhere. People consume them and they don't even know they are dealing with more or less the same drug when they take two or three or even more preparations of non-steroidals. So that's why you need to know the individual groups and the preparations. Again, I don't expect you to know the chemical group. I expect you to know the actual names of the drugs and the properties or the features associated with each, and it is very important. Anti-rheumatic drugs, now again, this part you hate very much, but again, only a few ones that I need you to know their names, but I'm interested that you know that there's something called disease-modifying drugs and drugs that treat the patient symptomatically without modifying the actual disease process. And among the disease modifiers, there's the targeted therapy, and there are two types of targeted therapies. There are the small molecules, and there are the monoclonal antibodies. And you need to know the mechanism of action or the mechanisms involved in the action of these drugs. Perhaps not the drug names. Perhaps you can refer to the drug list that I'm going to show in a while to tell you which names that you need to memorize. Otherwise, it's more about the classification according to being a targeted therapy, according to the disease ability to modify disease, and according to the mechanism of action. I've marked this in red or put it in red because I added something here, which is with special reference to targeted therapy. Uh, Again, I'm highlighting the important ILOs. Okay, you're going to be able to know which medications used for prophylaxis of acute attacks of gout that can precipitate or flare up, and the mechanism of this flare up is also very important. CNS stimulants, again, you need to know which region they work on in the brain, like what do brainstem stimulants do, cortical stimulants, psychotomimetics, analeptics. You need to know this classification, the meaning of these terms, and the use of each. The proposed mechanism of action of peptide hormones in okay, this regards doping. So doping is going to appear only in your end of year exam. And it's about actually the adverse effects, the mechanisms, and the possible methods. Not in too much detail. Now, I need you to understand about the minimum alveolar concentration of general anesthetics. And the ones I'm marking here are the most important. So please follow the marks of the eye on the ILOs. And again, with local anesthesia, I need you to know all about the amides, all about the mechanism of action, all about the factors that enhance or delay the action. And it's a bit tricky here because the local anesthetics, they need to work from inside the cell. Inside the cell, they need to be ionized to be able to work. But then to enter the cell, they need to be unionized. So that's why it's a little bit tricky. So anything that is going to increase acidity outside the cell is going to make them ionized extracellularly, and it's going to delay their action or inhibit their action or antagonize their action. But anything that's going to increase acidity inside is going to make them intracellularly ionized, and it's going to make them more potent, or is going to make their action stronger. So this is the trick here. All other factors, like the use of vasoconstrictors, like any other factor that uh, is associated with enhancement, prolongation of action, or the onset of action, all these factors are important. Uh, Again, for the individual ones, I need you to know about the amides. I need you to know about the adverse effects. I need you to know about the locally or the topically acting ester local anesthetics. I'm highlighting here the spectrum of penicillin because the spectrum comes in indirect questions. It doesn't tell you what's the spectrum, but it tells you a situation, and you know the situation is caused by a certain microorganism or a certain type of bacteria. So when you know the spectrum of that antibiotic, you know whether this is antibiotic is going to be good for treatment or not. So when you're studying the spectrum, uh, of course, on the marked ILOs, you study the spectrum because you know you want to apply it, you want to use it. You will not be directly asked about the spectrum. I can tell you confidently, there is not even a single question that's going to ask you whether this antibiotic is good for this type of bacteria. It's going to tell you whether it's good for this type of infection. Of course, Pseudomonas and anaerobes are an exception. We always ask about Pseudomonas, anaerobes, gram-positives, MRSA. So I'm sorry about that. Perhaps I can take it back. Yeah, spectrum can be asked when we talk about groups of bacteria that are important. So you need to know what's active against Pseudomonas, you need to know what's active against anaerobes, you need to know what's active against atypical bacteria, those without cell wall. You need to know generally what's good for the gram negatives and what's good for the gram positives, and within the gram positives, what's good for Staph aureus that is methicillin sensitive and Staph aureus that is methicillin resistant, and all the alternatives for this. All drugs that cover the gram positive organisms you need to know about, especially the resistant organisms. Of course, adding beta lactamase inhibitors to beta lactam antibiotics is a very important topic that you need to understand everything about. There's a tricky one here, which is called the enumerate the uses of carbapenems. There are no uses of carbapenems in your book, but here we are going to depend upon the spectrum. So we're going to treat infections caused by so and so and so and so organisms. This is going to be what we call the uses of carbapenems. Again, there is a difficult one here, which is mention alternatives to vancomycin for gram-positive coverage with characteristics of each alternative drug. We mean here MRSA and vancomycin-resistant Staph aureus and vancomycin-resistant enterococci. You need to know all about the gram-positive coverage. And to clarify this, I have also put it in red because I've added here that you need to know the glycopeptides, daptomycin, on one part of the book. And in another part of the book, there is talk about oxazolidine which are actually protein synthesis inhibitors. So it's basically in another chapter, okay, or in another part of the chemotherapy chapter, like linezolid and streptogrammin. This is also included in the gram-positive coverage as alternatives to vancomycin if bacteria are resistant. Tigecycline, you need to know all about it. Okay, and the warnings about it. Macrolides are a very important group. So as you can see, it's full of ILOs. TB, everything about TB is also important. But okay, how even if it's important to know which drugs are first line, this you have to know by heart. Some of the second line drugs you need to be able to recognize according to the drug list that I am providing. Also, you need to know how you give the anti-TB drugs. You need to know about the dot or directly observed therapy. You need to know about the drug interactions. You need to know about INH, what makes it more toxic, what makes it less, what makes it more toxic to the liver, what makes it more toxic to the nerves. This you need to know about. Antiviral drugs. This is a difficult topic, and I've added something here. It's very important. Please, whenever you are reading about antiviral drugs, always, always, always take note of how these drugs are going to be selectively toxic on virus-infected cells. How do these drugs target virus-infected cells in particular? Is there a special enzyme which is not there in the normal human cells that are not infected by viruses? Are these drugs targeting a certain structure of the virus which is not present in human cells? This you need to know about. Okay, so this is in addition to the previously mentioned ILOs. As you can see, virology is very important. Antivirals are very important. And again, not too many details apart from the drug names are mentioned in the book. So that's why it's a little bit tricky. I will try to make it simple. As for the antiprotozoals and antihelminthics, you're going just to go by the ILOs, and here the drug selection and drug choices are the most important. I hope this was informative. I hope this is going to help you through your exam, and I wish you the best of luck. I might not be answering as usual because you see, just before the exam, I need everyone to have the same chances. So forgive me if I cannot answer all your questions, but still feel free to send any questions and I will judge which ones can be answered individually and which ones cannot be. Thank you very much for your listening and I wish you the best of luck.